Hello Muhammad. Hello Muhammad. አይሰማም እስካሁን ያለው አትሰማን እስካሁን
እሺ አሁንስ ይሰማል?
አዎ ይሰማል
እሺ እሺ
እስካሁን አልሰማንም ነበር
በቃ እሺ ከድጋሚ እንጀምራለን። Okay, today's class it is about lipid metabolism. By this chapter, you are going to see or understand how dietary lipids are digested and absorbed to the body. It will be discussed, and then we will discuss about the beta oxidation of fatty acid, the energy production from fatty acid beta oxidation. It will be discussed. There are different types of beta oxidation: alpha, beta, and omega beta oxidation. Omega oxidation are there for energy production from this fatty acid. Now, this will be today's class. Yesterday, you are seeing the electron transport chain with Tensai. The electron transport chain is a continuation of lipid metabolism or lipid oxidation or fatty acid oxidation. Anyhow, you have taken it yesterday. So after beta oxidation, there will be energy production through Krebs cycle and electron transport chain. Unfortunately, you have taken it yesterday. After this, how endogenous fatty acids are synthesized, or de novo fatty acid synthesis, it will be discussed. Then after fatty acid synthesis, we will go to cholesterol metabolism, cholesterol synthesis and degradation or utilization by the body. It will be discussed. Finally, we will see the conversion of fatty acid into other products like arachidonic acid conversion to prostaglandin, leukotrienes, thromboxane synthesis. It will be discussed. So this is the whole picture. At the end, you will have question and answer. Let us start by having this digestion and absorption of lipid. Digestion, it have many functions in our body. Digestion, purpose of digestion is to convert one large lipid droplets, not only lipid, but large food particles are degraded or converted into smaller. As a result, we'll have easy absorption to the blood either or lymphatic vessel. In the case of lipid, it is to lymphatic vessel. The second function of digestion is to remove antigenicity of food, antigenicity, which can raise or
ሄሎ
sensitize.
መሐመድ፣
አቤት አይሰማም?
ስንተኛው ስላይድ ላይ ነህ?
ሁለተኛው ስላይድ
አዎ እኛ ላይ አንደኛው ስላይድ ላይ ነው ያለው።
እስካሁን
ምንድነው ስላይድ ሾው ስላደረከው ነው። ከሱ ወጥተህ ከሆነ ይሰራል።
እንደዚህ ይሁን።
ወጣ። በቃ እንደዚህ ነው።
እሺ main purpose of digestion it is to convert the size, number one. Number two is removing antigenicity and plus prevent allergy, which is come from external of our body. There are some allergic components that will be removed during this digestion process. And finally, we'll have absorption of important nutrients and excretion of waste materials through feces. It will be occur. The overview of lipid metabolism, the general overview, plus energy production or oxidation of fatty acid. It is shown here. First, there is lipolysis. Lipolysis means lysis of lipid or fat. It is occur by mechanically or enzymatically. It will be degraded into fatty acid, and then fatty acid enter into the beta oxidation. Through beta oxidation, it will be degraded to acetyl-CoA. Acetyl-CoA, it have 2 carbon. As you know, fatty acid, it have many numbers of carbon from 4 to 36 carbon. This is the length. After beta oxidation, every carbons of fatty acid, they will be degraded or chopped into acetyl-CoA. Acetyl-CoA is a common product of carbohydrate metabolism, protein or amino acid metabolism, and lipid metabolism. They form this acetyl-CoA. Acetyl-CoA now it can be join the Krebs cycle or citric acid cycle for the formation of reduced coenzyme. Either this acetyl-CoA will be used for cholesterol synthesis or cholesterologenesis. It is occur, and then steroids are formed. Steroids like different steroids are formed from this cholesterol, like steroid hormones, progesterone, estrogens, vitamin D formation, or other steroids are formed by this cholesterol molecule. The third one, this acetyl-CoA may join the ketone body synthesis, ketogenesis. It is occur. Ketone body synthesis, it is occur whenever this acetyl-CoA is formed in excess amount, and then this will join ketone body synthesis. So this is the general overview of fatty acid oxidation or lipid metabolism. Now let us start the digestion of dietary lipid in the mouse. First, let us see the main lipid items found in our food. There are 3 major lipid items in our food. The first one, or the major one, is triacylglycerol, TAG. This one is found in dominant form, which is about 95% of lipid contain triacylglycerol. Almost 100% of the lipid is made by a triacylglycerol. The remaining 5%, it is either free cholesterol or esterified cholesterol. The free cholesterol or esterified cholesterol, they make 10 to 15% of 5%. The other one, the rest one, it is made by a phospholipid. This phospholipid and cholesterol ester, they make the remaining 5%. The other one, the dominant one, it is made by this triacylglycerol, 95%. As you know from last time class, from module 1 or module 2, you know the structure of triacylglycerol. triacylglycerol, it have a glycerol backbone attached with 3 fatty acid at carbon number 1, carbon number 2, and 3. These are the 3 fatty acid attached with glycerol backbone. Cholesterol ester, it is a very complex molecule. It have fatty acid attachment at carbon number 3, steroid nucleus. This is the steroid nucleus, and the hydrocarbon chain, non-polar hydrocarbon chain, is attached at carbon number 17 of cholesterol. This is a cholesterol ester structure. Phospholipid, it have similar structure, but at carbon number 3, there is phosphate with X molecule. X, it is a variable molecule. Nitrogen base are attached at X position, and they make a different phospholipid. There are different phospholipids like phosphatidyl choline, phosphatidyl serine, ethanolamine. Based on the X molecule, will have a different phospholipids. So this is the general structure of phospholipid. So these are the 3 major types of lipid found in our food. The dominant one is made by this triacylglycerol, and the rest, the remaining 5%, are made by cholesterol ester and phospholipid. Digestion of lipid, it is begun in the mouth. By lingual lipase. The function of lingual lipase is not highly active like that of small intestinal digestion. This one is more active or more functional in the neonates than adult. In the neonate, it is highly active, but in adult person, this one, it is inactive. You don't have as much as function of pancreatic lipase. Lingual lipase is secreted from our mouth under our tongue. Ebner's gland secrete the lingual lipase and targets the short and medium chain fatty acid of the milk fats. Milk fats, after now, what you have to remember, fat means the triacylglycerol. Triacylglycerol, it will be degraded by a lingual lipase whenever this fat contains short and medium chain fatty acids. Short and medium chain fatty acids. Let me ask you one question. The short chain fatty acid to have how many carbons? What are the range of the carbon number found in short chain fatty acid? Do you remember? Do you remember the carbon number found in the short chain fatty acid, medium chain, long chain, and very long chain fatty acids? Tell me the range of carbon numbers found in short chain, medium chain, long chain, and very long chain fatty acids. This is very important because number one, when the carbon number is increasing, the solubility, it will be decreased. The absorption mechanism also, it is different. Here the enzyme also involved are different. Involved enzymes on the digestion of fatty acid, it is different. Lingual lipase primarily targets these fatty acids, the short and medium chain fatty acids. They will be targeted on the milk fat. Milk consumption is usually it is occur in the children or neonates. Adult person, the adult person, they are not milk consumers when they consume other types of food than milk. So this activity, it will be decreased upon the age, and it is highly active in the neonates. This enzyme again remove fatty acid at carbon number 1 and 3 of the triacylglycerol. It is not able to remove carbon number 2 of this triacylglycerol. Finally, at the end of this enzyme digestion, we'll have 2 molecules of free fatty acid and 2 monoacylglycerol are formed by this enzymatic digestion. The optimum pH for this enzyme, it is 4 to 6. 4 to 6, as you know, this is acidic pH. Acidic pH is found in the stomach than mouth. Already in our mouth, we have a neutral pH. In acidic pH is found in the stomach. So the enzyme of this enzyme function is occur in the stomach, even if it is secreted in the mouth. The function is occur or done in the stomach because its optimum pH is on the acidic side. After mouth, there is digestion in the stomach. The food bolus reach in the stomach. So in the stomach, we'll have another enzyme which is secreted from chief cell of the stomach called gastric lipase. Gastric lipase is our second enzyme act on the triacylglycerol again. This enzyme, they are similar like the first enzyme, lingual lipase. It is only acting on the short and medium chain fatty acid. It removes the short and medium chain fatty acid of milk fat. So this one needs emulsification or require bile salts emulsification in order to allow the digestion in the stomach. This emulsification function is given by polysaccharide, phospholipid, bile salt. They give bile salts is secreted into small intestine, but in the stomach we have this polysaccharide and phospholipids. Plus protein, they give emulsification function in the stomach. The optimum pH again, it is found between 4 to 6. This is again, it is an acidic pH found in the stomach. It will give us optimum pH or optimal working condition for this gastric lipase enzymatic digestion. Okay, for example, the acidic condition in the children and the acidic condition or the acidic pH in the stomach of adult person is different. Adult person, in adult person, the acidic or the stomach to have strongly acidic pH, which is found between 1.6 to 3.2. But in the children, the environment of acidic, the environment of stomach is not acidic like adult person. So this enzyme is more functional in the neonate or in the children than adult person, similar like the first enzyme called lingual lipase. After this enzymatic digestion, we'll have again 2 molecules of free fatty acids at carbon number 1 and 3 of triacylglycerol are removed, plus 2 monoacylglycerol is formed by this enzyme. This will be the products of gastric lipase enzyme digestion. So here in the stomach, whenever the food or fatty food arrived in the stomach, cause the release of different hormones like cholecystokinin. It is a hormone which stimulates the contraction of pancreas and contraction of gallbladder. Secretin hormone, this one again stimulates the release of insulin. All these three, they stimulate the release of insulin, gastric inhibitory peptide, glucagon-like peptide 1. They stimulate the release of insulin upon food consumption. Okay, these 4 hormones, they are secreted upon the arrival of food to the stomach or small intestine. Chyme enter in the small intestine, make the release of these 2 hormones, cholecystokinin and secretin, plus the other hormones, they will be secreted upon food arrival. Functions of cholecystokinin, this one is very important to stimulate the pancreatic enzyme secretion upon this contraction, and gallbladder contraction, it will be stimulated by this cholecystokinin. As a result of this, gallbladder release bile cell to small intestine, pancreas release proenzymes or zymogens. They will be released to small intestine, and then we'll have digestion by this enzyme. Secretin is important to stimulate the contraction of, again, pancreas. So this pancreas, it is an accessory organ for this digestive system. Gallbladder also store bile cell, which is synthesized in the liver, and finally release bile cell into small intestine. So the first process in the small intestine, it is emulsification function. Emulsification process, it will be occur before any enzymatic digestion. This process, it will be occur. Emulsification, this one, it is important to increase the interaction between enzyme, increase the surface area of this hydrophobic fat droplet to the digestive enzyme. As a result, we'll have maximum digestion function from these enzymes. So this one emulsification function, it is look like detergent. The detergents, they will remove the oily dirt like after you consume the fatty diet, that will be washed off from this our plates using this detergent. So these bile cells act as a detergent and make interface, allow the interaction between a polar and non-polar molecules. Polar and non-polar, they will be interacted as a result of this emulsification function. This will be given by number one, peristaltic movement, mechanical mixing. It is occur upon the movement of the ball, small intestinal ball. Bile cell, it is a major one. This will be very important to give this emulsification function. It is act as a detergent or detergent properties will be given by these bile cells. As a result, it will decrease surface tension between lipid droplets. It will decrease the surface tension and there will be disintegration of lipid droplet. It is occur as a result of bile cell. Phospholipid again, it is anamphipathic. It will have polar and non-polar parts. Using polar parts, it will interact with the polar solvents. Non-polar part binds to the lipid part or fats. As a result, we'll have interaction between polar and non-polar using this mediator. This is it is act like an adapter between polar enzyme or polar environment with non-polar lipid. All this they give us, they will give us emulsification. As a result, enzyme will get interaction or interface between lipid and enzyme. It will be occur. So we'll have maximal digestive function. It will be occur as a result of this emulsification function. Okay, let us see types of bile acid, the sites where it is incised, plus the storage location and types of bile acid. It will be it is very important. As you know, bile acid, it is made in the liver from cholesterol, either endogenously synthesized or from exogenous source. We'll have cholesterol, and then this cholesterol, it will be used for the synthesis of bile acid. Cholesterol structure, you know, the cholesterol structure, it have steroid nucleus with one OH group, with one alcohol functional group at carbon number 3 only. But after conversion of after bile acid formation, we'll have many hydroxyl groups. There are several hydroxyl groups on the steroid nucleus. So this will give us increase the polar part of bile acid. It will be increased upon the addition of more number of hydroxyl group on the steroid nucleus. So this hydroxyl group it will be added on the steroid nucleus. This will increase the polarity of cholesterol molecule. After it is synthesized, it will be stored in the gallbladder. Whenever there is food arrival or fatty diet in the small intestine, it will be secreted into small intestine. There are two types. One of bile acid, it is called cholic acid. We call it cholate. This will be attached or conjugated with amino acid like glycine amino acid. It is added. Plus the other type, it is taurine. Taurine, it is derived from cysteine amino acid. Cysteine amino acid, it is added. Cysteine, it is converted into taurine, and then it will be added to these cholates and form taurocholate. It will be formed. Glycine amino acid addition form a glycocholate bile acid. Still, they are bile acid. Bile salt formation, the salt form, it is formed after the addition of ions like sodium or potassium. They are added. Potassium addition like potassium glycocholate or sodium glycocholate, they will be formed after the addition of ions. Sodium from sodium hydroxide or potassium from potassium hydroxide, they will be reacted for the formation of the salt. The second type of bile acid, it is called chenodeoxycholic acid. Chenodeoxycholic acid, it is the second type. Again, similarly, we add glycine amino acid or taurine on the chenodeoxycholic acid for the formation of kenodeoxycholic glycokenodeoxycholic acid. It is formed after glycine is added. Taurokenodeoxycholic acid is formed after the addition of taurine. So this is one type, and the second type, it is called kenodeoxycholic acid. Don't forget, there are two types. So they will give us emulsification function, and then we'll have interface between lipid and enzyme. This is how it works. We have a large lipid droplets before emulsification. This one is, they are aggregated each other to avoid water contact. They aggregate each other. But after bile acid or salt addition, this large lipid droplet, it will be disintegrated into smaller lipid droplets. The outer part of these lipid droplets, it is covered by bile salts, bile acid, phospholipid. They cover the outer part and make soluble malt. This one, it will be, it can be circulated, or even it can be soluble in water. So the enzyme finally acts on the lipid part, having this emulsification function. It is very important. So without this bile acid, we cannot digest our lipid diet. So it is very important to have maximal lipid digestion after this emulsification function. The enzymes which are secreted from pancreas for lipid, there are 3 different types of enzyme. Number 1, there is pancreatic lipase enzyme. This one, it is secreted from pancreas, released from pancreas into the first part of small intestine, duodenum, jejunum, and ileum. These are the 3 parts of small intestine. The first part, it is called duodenum. We'll have pancreatic lipase enzyme digestion on the first part of small intestine. Pancreatic lipase, it is for the degradation of triacylglycerol. Triacylglycerol, it will be degraded by this pancreatic lipase enzyme. This enzyme, it is activated by calcium and colipase enzyme. They activate pancreatic lipase. Bile salt inhibits pancreatic lipase enzyme. So the activity of this enzyme, it will be inhibited by bile salts and activated by colipase enzyme and calcium. Here is the enzyme. Colipase and calcium, they activate bile, inhibit the enzyme activity. Again, this enzyme, it is able to remove carbon number 1 and carbon number 3 of the triacylglycerol, finally produce 2 molecules of free fatty acid here, and 2 monoacylglycerol. The carbon, this number 2 indicates the location. This carbon number 2 fatty acid, it is not able to, it is not removed by pancreatic lipase. So it will be formed. It will be remained as a 2 monoacylglycerol after pancreatic lipase digestion. So we have these 2 molecules of free fatty acid and 2 monoacylglycerol. This one indicates the carbon location, not number of molecule. Okay, 2 monoacylglycerol. It is after this digestion, we'll have a 2 monoacylglycerol formation. Digestion of cholesterol, cholesterol ester, it is done by the second type of enzyme called cholesterol esterase enzyme, which is again released from pancreas. It will act on cholesterol ester and produce cholesterol, free cholesterol, and free fatty acids are formed after esterase enzyme degradation. Again, this one is the activity of this enzyme. It will be increased in the presence of emulsification by this bile salts. The carbon number 3 fatty acid, it will be removed by this esterase enzyme. This one produce free fatty acid, free cholesterol, and fat-soluble vitamins like vitamin A, vitamin E, vitamin D. This will be formed by a cholesterol esterase enzyme. Okay, these are they are fat-soluble vitamins. Vitamin A, E, D, and K. They are fat-soluble vitamins. They will be formed after esterase enzyme degradation. The third enzyme called phospholipase A2. Phospholipase A2, it is formed as or released as a proenzyme. Proenzyme means inactive. Enzymes are secreted or released into small intestine. Another name of this proenzyme, it is zymogen. The term zymogen you have learned, I think, before during protein digestion or carbohydrate digestion. There are a proenzyme secretion into small intestine to avoid self-digestion plus to have safe transport and storage. So they are secreted as inactive form. They will be activated by, for example, this one is activated by trypsin. Trypsin is enzyme for protein digestion, and it is important to activate phospholipase A2. Again, the enzyme require emulsification function by bile cells, remove fatty acid at carbon number 2 of phospholipid. Phospholipid, this is the structure of phospholipid. There are two fatty acid attachment at carbon number 1 and 2. Carbon number 3, it is attached with phosphate and X molecule. The enzyme phospholipase A2 able to remove carbon number 2 of fatty acid. Okay, this will be removed by phospholipase A2. There are other isoenzymes. Isoenzymes, they remove fatty acid at different carbon number. A1, phospholipase A1, it is able to remove carbon number 1 of a phospholipid. Phospholipase A2, it is able to remove carbon number 2 of a phospholipid. Phospholipase C, it is removing the carbon number 3 of phosphate. The nitrogen-based X molecule, it is removed by phospholipase D. But in our intestine, or pancreas secretes or release this enzyme. Phospholipase A2 is the one secreted into small intestine. A1, phospholipase C, and phospholipase D, they are found in the cell inside the body or in the target organ. We'll have this A1, C, and D. For example, phospholipase C, it is important to secrete or form a secondary messenger. Secondary messengers, they are formed as a result of activation of phospholipase C in the membrane of the cell. This is one example. So they are found in the cell of our body, in different parts of our body. They are found in the intestine. We have this isoenzyme. Phospholipase A2 is the one found in the small intestine, which is released by this pancreas. So at the end of this enzyme digestion, we'll have one free fatty acid formation, and lysophospholipids will be formed. Free fatty acid plus lysophospholipid will be formed at the end of this enzyme digestion. So we finished all the three types of lipids. Now they are digested by enzyme. Pancreatic lipase for triacylglycerol, cholesterol esterase is for cholesterol ester, phospholipase A2 it is for phospholipid digestion. They digest their own types of lipids. Finally, at the end of this lipid digestion, we'll have end product formation. For example, the end products are free fatty acid, 2-monoacylglycerol, cholesterol, and lysophospholipid will be formed at the end of this enzymatic digestion. So first, there is a dietary lipid consumption. In the mouse, hardly there is digestion by lingual lipase, cholesterol ester, phospholipid, triacylglycerol. They are unchanged. We don't have that much function in the adult person, so it will be insignificant. In the stomach also, we have a minimal digestion by gastric lipase. It is more active in the neonates or children. So most lipid digestion is occur in the small intestine with the help of pancreas. Pancreas secrete number 1 proenzymes. They will be secreted into small intestine. Number 2, bicarbonate is secreted. Bicarbonate, as you know, it is important to neutralize the acidic pH of stomach. Acidic pH of stomach, it will be neutralized and then make alkaline pH in the small intestine. As a result, we'll have optimum pH for these pancreas enzymes. Gallbladder secrete bile salts into small intestine through bile ducts. Through bile ducts, there is secretion of bile salt into small intestine. Then we'll have emulsification function in the small intestine. To the end of small intestine or at the ileum part, there will be absorption into enterocytes. This is the lumen part of our intestine, and this is the enterocytes. Enterocyte of enterocyte or the mucosal cell of small intestine. They will absorb the dietary food, dietary lipid into enterocytes. So choline, here this one is a nitrogen base found in the phospholipid. Okay, this is the structure of cholesterol ester. It is acted by cholesterol esterase into cholesterol plus free fatty acid. Fatty acid, it will be removed at carbon number 3. This is a phospholipid structure, phospholipid. There are 2 fatty acids at carbon number 1 and 2. At carbon number 3, there is phosphate with nitrogen base choline. It is acted by a phospholipase. They remove fatty acid at carbon number 2 plus carbon number 1. Sometimes carbon number 1 also it will be removed, and then we'll have a glyceryl phosphoryl choline. It is formed plus 2 free fatty acid. But the major or the default one is phospholipase A2 act on carbon number 2 and form 1 free fatty acid and lysophospholipid. Triacylglycerol by pancreatic lipase remove carbon number 1 and carbon number 3 of this triacylglycerol and form 2 molecules of free fatty acid. 2 molecules of free fatty acid plus 2 monoacylglycerol are formed by the action of pancreatic lipase. So these are the 3 types of lipid released from pancreas and then digest our dietary lipid. So after we have this digestion, there will be absorption into lymphatic vessel first. These end products, they will be absorbed into lymphatic vessel. First, enterocyte. In the enterocyte, there is re-esterification, re-synthesis of triacylglycerol, re-synthesis of cholesterol ester, re-synthesis of phospholipid. It is occur in the enterocyte. After making those lipids, protein, it will be added. Proteins are added for the formation of the first lipoprotein, chylomicron. It is formed. Then it will be delivered or travel through this lymphatic vessel through our vena cava, superior vena cava. It will join to the blood circulation. Okay, it will enter into the blood circulation. Through the blood circulation, there will be metabolism of chylomicron. It will be occur. So we will discuss how it is metabolized later. Let's see the detailed mechanism of lipid absorption into intestinal mucosal cell. At the second part of small intestine, the jejunum, we'll have complete lipid digestion. Finally, we'll have this end product, free fatty acid formation from those lipids, free cholesterol formation from cholesterol ester, 2 monoacylglycerol from triacylglycerol, lysophospholipid from phospholipid. They are formed, and this will be combined with bile and fat-soluble vitamins for the formation of micelle. Micelle will be formed. This is the micelle structure. Micelle, it is formed again with bile. Okay, there is bile addition on the end products of lipid digestion, and then we'll have this micelle formation. Micelle, it is look like cell. There is cell membrane on the outside, but the cell membrane it is made by bile cell, not phospholipid. This make the outer layer non-polar molecules. They will be found inside or avoid water contact, and they form this micelle. Micelle is formed. Now this micelle, it will be absorbed passively into mucosal cell, soluble in aqueous intestinal environment and absorbed at the brush border of enterocyte into inside of this enterocyte. It will be absorbed passively without energy consumption. What you have to notice, the short and medium chain fatty acids or the carbon number between 4 to 6. Carbon number between 4 to 6, they are called medium short chain. From 8 to 12, they are called medium chain fatty acids. They will be absorbed directly into portal vein, not to the lymphatic vessel. They are soluble in water, short and medium chain fatty acid. They are soluble in water, therefore they can directly absorb it into portal vein. So there is no need to micelle formation for this short and medium chain fatty acid. Then after micelle formation, there is a passive absorption to the enterocyte. Again, this is another explanation. By this picture, we can see how re-esterification is occur. We have digestion, then there is absorption into enterocytes. After absorption, there is re-esterification of the end products of lipid into triacylglycerol, cholesterol ester, and phospholipid. The three lipids, they will be re-synthesized. And then we add proteins, apolipoprotein B48, apolipoprotein A1, they will be added to this lipid, triacylglycerol, fat-soluble vitamins, cholesterol ester, and phospholipid on the outer parts. They make this lipid droplet with the addition of protein. Here we add protein, and they make a chylomicron. Chylomicron, it is our first lipoprotein, which transport exogenous lipid, dietary lipid. It is transported by using this chylomicron. Now this will be secreted into lymphatic vessel. Through the lymph, it will travel up to superior vena cava. In the superior vena cava, it will be delivered or drained to the blood circulation. Absorption of fat into enterocyte is mainly done through diffusion. Diffusion means passively from high concentration area to low concentration area. By this diffusion, we make low concentration. You see, this re-esterification is required to make this lipid droplet, lipid end products low in concentration. So after absorption, they are re-esterified. As a result, we'll have a low concentration. Then this will allow the passive absorption or diffusion. It is allowed as a result of this re-esterification process. Fatty acid transporter or CD36, they transport or they carry the lipid end products into enterocyte by this transporter molecule found in the cell membrane of small intestine. Re-synthesis of triacylglycerol. Step by step, we add fatty acid on the glycerol backbone. Before that, we have to have activated fatty acid. The free fatty acid, it is activated into fatty acyl-CoA in the enterocytes by using fatty acyl-CoA synthetase enzyme. This enzyme, it is energy-consuming enzyme. It requires 2 ATP. One ATP is it is hydrolyzed to AMP. Therefore, we use 2 high-energy phosphate bonds. The 2 high-energy phosphate bonds are used for the activation of one fatty acid into fatty acyl-CoA. So this is equivalent with 2 ATP consumption. It is equivalent with 2 ATP consumption. So fatty acyl-CoA synthetase enzyme activates fatty acid into fatty acyl-CoA. Once it is activated, it can esterified with glycerol or alcohol functional group for the formation of triacylglycerol. So the 2 monoacylglycerol attached with 2 fatty acyl-CoA for the formation of triacylglycerol. So each fatty acid, they are attached one by one, not at a time. They are attached or esterified one by one. First, one fatty acid is added and diacylglycerol is formed, and the third one is added for the formation of triacylglycerol, which is the last one. Okay, there will be activation first, then after activation, it will be esterified for the formation of triacylglycerol. So this one is triacylglycerol synthesis pathway. It is re-synthesized in the enterocytes. Phospholipid and cholesterol ester re-synthesis. It is occur again by adding fatty acid on the cholesterol for the formation of cholesterol ester. Again, we add fatty acid to lysophospholipid for the formation of phospholipid. They do have their own enzyme. For example, for cholesterol ester formation, there is acyl-CoA cholesterol acyl transferase enzyme transfer fatty acid to cholesterol for the formation of cholesterol ester. Reacylation of lysophospholipid is caused by acyl transferase enzyme. Acyl transferase enzyme transfer fatty acid for the formation of phospholipid. Okay, acyl-CoA, acyl transferase, it is for cholesterol ester. Acyl transferase enzyme, it is for the formation of phospholipid. Using these two enzymes, there will be resynthesis in the enterocyte. So we have three of the lipid in the enterocyte by these two enzymes. There are two isoenzymes for acyl-CoA, acyl transferase. Acyl-CoA, acyl transferase 1 and 2. Their expression is different at different cell. Plus, they by their unique physiological function, they are different. Acyl-CoA, acyl transferase 2, it is mainly expressed in the liver and intestine. The other one, acyl-CoA, acyl transferase 1, it is present in most tissue. Most tissue have this enzyme. So acyl-CoA, acyl transferase isoform, again, they are found in the macrophage and the steroidogenic tissues. They contain this enzyme for making cholesterol ester from free cholesterol and fatty acid. Here you see the free fatty acid with short and medium chain lengths. They are directly absorbed to the portal circulation. Portal vein will absorb the short and medium chain fatty acid. The reason for this, they are soluble. I have said it before. So the short and medium chain fatty acid, they are soluble in water. They don't need to form micelle, directly absorbed into portal vein. Glycerol again diffuse to portal vein and then reach in the liver for the formation of glucose. From glycerol, there is glucose formation through gluconeogenesis. Gluconeogenesis convert glycerol to glucose either, or to make another triacylglycerol, we use this glycerol backbone. Again, summary of resynthesis of lipid in the enterocytes. There is two monoacylglycerol. This one, at a time, one fatty acid is transferred for the formation of diacylglycerol. Then triacylglycerol is formed. The long chain fatty acids, they are activated first using this CoA attachment, CoA addition, plus ATP is consumed by this enzyme, and the fatty acid is activated into fatty acyl-CoA. Once it is activated, it is added to this to monoacylglycerol, and triacylglycerol is formed. Cholesterol again is esterified by adding one fatty acid for the formation of cholesterol ester. Phospholipid also, it is formed by the addition of one fatty acid. So chylomicron is formed when we add amino proteins. This one is unique for chylomicron. Apolipoprotein B48, it is unique for this chylomicron. Do you know the function of this protein part? What are the functions of the protein part in the lipoprotein? Do you have any suggestion? Class activity ያስፈልጋል አይደል? Class activity, what will be the major functions of protein in the lipid part or in the lipoprotein? Do you know? እሺ አንሚዩት መሆን ትችላላችሁ መልስ ለመመለስ። ቻት ማድረግ ይቻላል። የስ ትክክል። አንደኛው ሶሉብላይዝ ነው የሚያደርገው ወይም ትራንስፖርት ኢንሶሉብል ሊፒድ ስሩ ዘ ብለድ ሰርኩሌሽን። ትክክል። ሌሎቻችሁም ፓርቲሲፔት ብታደርጉ ጥሩ ነው። there is the protein part, it is important. Number one, solubilization. There is solubilization of non-polar lipid in the blood circulation. So the lipid, they can transport it through the blood circulation. As you know, blood, it is aqueous. It is water soluble. Therefore, the non-polar or insoluble lipids. To use this blood circulation, we have to solubilize this lipid. So we add this protein to make the lipid is soluble in the blood or in the water solution. Number one. Number two is it is to activate enzyme. At the end, this chylomicron enter into the blood circulation and reach in the target organ, and then it is hydrolyzed or it is degraded by enzyme. Those enzymes, they are not active always. They are activated by proteins found here or surrounding this chylomicron. It is for activation of hydrolyzing enzyme in the blood circulation. Number two. Number three is to recognize receptor, recognition of receptor. It is occur. Serve as, yes, ligand. Yes, serve as a ligand for lipoprotein interaction. ትክክል ነው። አንደኛው ኢንዛይሞችን አክቲቬት ለማድረግ ነው። activate the enzyme. Number two, it is to solubilize this lipid. Number three is to recognize the receptor or act as a ligand and bind to the receptor, especially to the liver receptors. They bind to this liver receptor. As a result, there will be uptake of lipid into enterocyte. Number four function, integration when maintain the composition or maintain the integrity of this lipoprotein is occurred by this protein part. So they are the functions of the protein part in the lipid. Absorption of bile cells. Bile cells, it is derived from cholesterol. ስለዚህ if this cholesterol, if this bile cell is excreted, then indirectly we excrete cholesterol from the body. If it is reabsorbed, we are reabsorb cholesterol into the body. After chylomicron is absorbed or the end product of lipids are absorbed into enterocyte, bile remain behind. So at the end of the small intestine, it will be reabsorbed. At the end means at the ileum part of distal ileum part of small intestine, it will be reabsorbed into enterocytes. Most part it will be reabsorbed, but few it will be excreted to the feces. Okay, bile cells are not absorbed together with products of hydrolysis of dietary triglyceride, phospholipid, cholesterol ester. They are not incorporated into chylomicrons in the enterocytes. Instead, bile cells remain in the intestinal lumen until they reach distal ileum, where most are absorbed by active transport mechanism that utilize sodium bile acid or bile cell co-transport. So it is sodium dependent. With sodium, it will be transported into enterocytes. Sodium, it makes active. Sodium dependent means it is active. So the sodium amount, it will be adjusted, or concentration, it will be adjusted by using sodium potassium pump or sodium potassium ATPase channel. Adjust the sodium amount inside low to make inside low. As a result, bile cells, it will be transported into enterocytes. Sodium potassium pump consumes the ATP, and this makes active absorption, or bile cell absorption is active indirectly, not direct ATP consumption, but indirectly it is active because the ATP consumption, it is occur by the sodium potassium pump, not by sodium bile cell absorption mechanism, or import of sodium and bile cells will not consume ATP. The disorder in lipid metabolism or lipid digestion and absorption, sometimes there may be different diseases. Upon these different diseases, we'll have excess fat secretion or excretion through feces. In that case, we'll have steatohorrhea. This one, it is occur whenever we have problem in the intestine, either or pancreas or gallbladder, bile cell secretion deficiency, bile duct obstruction, or due to pancreatitis or pancreatic cancer. They cause this steatohorrhea. Intestinal disease, different intestinal diseases also cause steatohorrhea. So this is a disease state. Whenever we found excess lipid in the stool, we call it steatohorrhea. Increased lipid and fat-soluble vitamins excretion in the feces called steatohorrhea. Normally, in the normal person, we have less than 5 grams per day fat in the stool. But in the case of steatohorrhea, there will be more amount of fat in the stool that is greater than 5 grams per day. It will be excreted through feces. This one is caused by defects in lipid digestion or lipid absorption anywhere in the intestine or in the accessory organ. Defect cause this steatohorrhea. Secretion of lipid from enterocytes. Secretion after absorption into enterocytes, there will be secretion into lymphatic vessel, lymph vessel. Before secretion, there is formation of chylomicron. Chylomicron is formed by the addition of proteins. We said before, there is aggregation of those lipid, triacylglycerol, cholesterol ester. They will be found in the center because they are non-polar. Triacylglycerol and cholesterol ester, they will be cargo, where they will be found in the center of chylomicron. Polar or amphipathic molecule, they will be found on the outer surface. For example, phospholipid, it have a polar part. Cholesterol, the free cholesterol, it have one alcohol functional group. Therefore, it have a small polar head that will be found on the outer part of this chylomicron, and plus proteins, different proteins, apolipoprotein B48 and apolipoprotein A1. They will make the outer surface. So this is a chylomicron. This will be formed in the enterocytes. After making this enterocyte, this will be it can be released to into lymphatic vessel or lactate by exocytosis. Exocytosis and endocytosis. Endo means uptake or phagocytosis. It have two types: exocytosis and endocytosis. Releasing, it is exocytosis, and uptake or taking, it is endocytosis. After lipid-rich meal, the lymph become chyle or milky appearance will be occur after lipid diet, digestion and absorption and secretion into lymphatic vessel. From lymph, chylomicron finally enter into the blood circulation. They join the blood circulation. So in the blood circulation, again, it will be matured. በነገራችን ላይ chylomicron formed in the enterocytes, it is immature or nascent chylomicron. We call it a nascent chylomicron or immature chylomicron. After it is enter into the blood circulation, it will be matured by the addition of more proteins. More proteins will be added to chylomicron and then makes chylomicron matured. So assembled in the intestinal mucosal cell, it is the lowest in density, largest in size, the highest percentage of lipid, and the lowest percentage of protein is found in the chylomicron. There is protein in lipid, but the lipid part is the highest and the protein part is the lowest in percentage. From the lipid part, triacylglycerol is making the highest proportion. It carry exogenous lipid or dietary lipid to periphery of our body, and it is responsible for physiological milky appearance of plasma up to 2 hours after meal. So this one is, it is properties of chylomicron. So this is a graph. It shows us the weight, the percent of total weight in the different in chylomicron only. Okay, chylomicron, it have this component or compositions. Triacylglycerol make the highest composition, or it makes the largest weight in this chylomicron. Protein parts, the free cholesterol, cholesterol ester, and phospholipid, again, they are found in minor or in small amount than triacylglycerol. Okay, this one is chylomicron. Chylomicron, it is the largest in size and the smallest in density. Okay. Metabolism of chylomicron in the blood circulation. The nascent chylomicron here, nascent or the immature chylomicron are matured. They will be matured by the addition of further proteins like ApoC2, apolipoprotein C2, and apolipoprotein E are added to this chylomicron by this HDL. HDL donate those protein to the nascent chylomicron, and this one become matured. Chylomicron become matured after accepting apolipoprotein to this chylomicron. Let's see the functions of those protein. Apolipoprotein B48 is for transport function, solubilize the non-polar lipids, and then transport to the blood circulation. ApoC2, apolipoprotein C2 is important to activate lipoprotein lipase enzyme found in the blood circulation, in the blood vessel. Apolipoprotein E is to bind liver receptor. Okay, this one acts as a ligand and binds to liver receptor. So we'll have this protein addition by this HDL lipoprotein, HDL cholesterol, high density lipoprotein. Cholesterol add or donate this to protein to this nascent chylomicron, then it become matured. Okay, activate the enzyme lipoprotein lipase enzyme. So lipoprotein lipase enzyme degrade triacylglycerol in chylomicron. This will be degraded by lipoprotein lipase enzyme in the chylomicron, and it form a free fatty acid and glycerol by this enzyme. Free fatty acids, they will be taken by either metabolically active tissues like muscle. Muscle, it can take fatty acid to have energy production or oxidation of fatty acids. Through this, it supply energy. Glycerol, it will be used for gluconeogenesis, for glucosynthesis, or it will be bind with another fatty acid for making another triacylglycerol. This is one option or one phase. The second phase, fatty acid, either it will be stored in adipose tissue. Adipose tissue is the area where lipid is stored. Fatty acid with glycerol, they are conjugated for the formation of triacylglycerol and then stored in the adipose tissue, white adipose tissue, not brown. There are two types, brown and white adipose tissue. The white one is for storage. The brown is for thermogenesis or heat generation. It is occurred by the brown adipose tissue. Okay. The lipoprotein lipase enzyme, it is found. This one is found in the blood vessel. This is the blood vessel wall or the endothelium of the blood vessel. We have this different polysaccharide chain. The polysaccharide chain, they attached this enzyme, or they have a lipoprotein lipase enzyme. You see, this is the enzyme attached with endothelium of the blood vessel. It is immobile or fixed in one position. Whenever the chylomicron pass through this blood circulation, the ApoC2 of chylomicron activate the enzyme and act on triacylglycerol. The triacylglycerol, it will be degraded by this activated lipoprotein lipase enzyme into free fatty acids and glycerol. Okay, this is occur. After we release fatty acid from this chylomicron or triacylglycerol, the free fatty acid will be taken by for energy production in the muscle or storage in adipose tissue. Extracellular enzymes anchored by heparan sulfate. This one is a complex carbohydrates, heparin or heparan. Polysaccharides. There are two types of polysaccharide, as you know. One is homopolysaccharide and heteropolysaccharides. Homopolysaccharide, they are made by the same monosaccharide. The same monosaccharide, they make the polysaccharide, like glycogen, starch, they are made by glucose only. Cellulose, again, it is made by glucose only. One type monosaccharide is found. Heteropolysaccharides are made by more than one sugars. This is one example. Heparan sulfate, it have more than one sugar. more than one type of sugar. It is found in the heparan sulfate, and this one attached or bind lipoprotein lipase enzyme, and it will be activated by POC2 for the degradation of triacylglycerol. This one is predominantly present in adipose tissue, cardiac, and skeletal muscle to release this fatty acid and use fatty acid as a source of energy. For its activation, it requires ApoC2. So ApoC2 activates the enzyme, then there will be degradation of triacylglycerol, degrade triacylglycerol into glycerol and free fatty acid. Insulin stimulates its synthesis and transfer to this luminal surface of capillary tissue, capillary blood vessel. During starvation activity, it will be declined in adipocytes and increased in cardiac muscle. Okay, this is very important. How it is regulated also is indicated by this sentence. It is very clear. Metabolism of chylomicron. First, it will be acted by a lipoprotein lipase enzyme. After, it is activated by ApoC2, and then remove the triacylglycerol. ApoE, it is important to bind liver receptor. Liver cell recognize ApoE and take the remnant chylomicron. Remnant chylomicron rapidly taken by this liver cell. Okay, in the liver cell, there will be further degradation. It will be acted by lysosomal enzyme and degradation of all the components. It will be occur. As a result, release the amino acid from the protein part, free cholesterol, and fatty acid. They will be released from this remnant chylomicron in the liver cell. The receptor is recycled. Okay, after phagocytosis or engulfing receptor into lysosome, the receptor they will be recycled or expressed on the cell surface after this process. So ApoE is for binding liver receptor and taking the remnant chylomicron. ApoC2 is activation of lipoprotein lipase enzyme. As a result, we'll have triacylglycerol hydrolysis. The remnant chylomicron, it will be taken by liver for complete degradation. So this is the complete picture now. It shows us the transport of lipid through the lymphatic vessel and blood circulation. Finally, in the blood circulation, how it is degraded by lipoprotein lipase is shown by this diagram. So after food digestion and absorption in the intestine, there is chylomicron formation, and this one is secreted into lymph. The lymph finally drain its important component into the blood circulation. In the capillary or at the periphery of our body, this chylomicron, it will be activated by ApoC2 or HDL mature the chylomicron, and LPL, it will be activated by ApoC2. The activated LPL will degrade triacylglycerol into free fatty acid and glycerol. Fatty acid, either it will be used for energy production, generation of energy, or it will be stored in adipose tissue. Triacylglycerol will be formed and stored in adipose tissue. The remnant chylomicron will be taken by liver by using this receptor. Receptor recognize ApoE in the chylomicron and take ApoE into liver cell. We'll have hydrolysis in the liver. So lysosome, as you know, it is hydrolytic or suicidal bug. It have hydrolytic enzyme. After phagocytosis of this engulfed particle, there is fusion with lysosome, and lysosomal enzyme degrade chylomicron into free fatty acid, cholesterol, amino acid, and glycerol. This will be formed. The receptor, it will be recycled. It will be recycled, and it will be re-expressed on the surface of liver. Use of dietary lipid by the tissue. The fate of fatty acid after it is removed by lipoprotein lipase enzyme, it will be oxidized by various tissues for energy production, and it will be stored. The second fate, it is storage. Fates of glycerol used by liver to form glycerol 3-phosphate, which can enter into glycolysis or gluconeogenesis. Through glycolysis, there is two generation or two net ATP will be formed. Through gluconeogenesis, it will be converted back to the glucose. So this will be a source of glucose synthesis during hypoglycemia. Hypoglycemic condition cause the conversion of glycerol to glucose. So gluconeogenesis and glycolysis, they are simple reversal. They are simple reversal, except some reactions. Out of the 10 series of reaction, 3 of the reaction they are irreversible. The rest 7 are reversible. So we can use those reversible reaction during glucose. Other sources. Do you know any other source of glucose in the gluconeogenesis? Okay, other source in gluconeogenesis, source of glucose in gluconeogenesis, number 1, amino acid, lactate, pyruvates, it's correct. Amino acid, glucogenic amino acids can be converted to glucose, lactate, pyruvates, propionyl-CoA, propionyl-CoA, which is formed by beta oxidation, odd number carbon fatty acid oxidation, form propionyl-CoA, glycerol from lipolysis, or this degradation of lipolysis or degradation of triacylglycerol form a glycerol. So these are some of the source of glucose through gluconeogenesis. Gluconeogenesis, it is activated or stimulated during hypoglycemic condition. Glycolysis, it is activated whenever we are in hyperglycemic condition. It is activated and oxidize glucose for energy production. Let's have some break. After 50 minutes, we'll rejoin. Do you have any questions still? እስካሁን በነበረው ላይ ጥያቄ ካላችሁ መጠየቅ ትችላላችሁ። ብሬክ እንውሰድና 15 ደቂቃ በኋላ እንገናኝ። 15 ደቂቃ Okay, thank you very much. After break, we will discuss about oxidation of fatty acid. I think this will be the first fate of fatty acid in our body after food consumption or food intake. The fatty acid either it is come from exogenous, from dietary lipid, or come from endogenous lipid, which is synthesized in the liver organ. Okay, after fatty acid is oxidized, there will be high amount of energy production than even glucose. Glucose, as you know, it is our primary source of energy in our body. It gives us some amount of energy will be obtained upon oxidation of glucose. But fatty acid, upon the oxidation of fatty acid, will have more number of ATP than carbohydrates. The reason for this one, fatty acids, they are highly reduced form. They are found in low oxidation state than hello.
Hello.
አቤት
ፓወርፖይንት የለም ይታያል።
ኦኬ እሺ ስለዚህ እንደቀደሙ ስላይድ ሾውን አጥፍቼዋለሁ። እሺ ፋቲ አሲድ ኦክሲዴሽን
አይደለም፣ ምንም ፕሬዘንቴሽን ይታየን አይደለም፣
ምንም አይታይም።
ሼር የተደረገልን አይደለም።
እሺ አሁንስ ይታያል?
ይታያል።
እሺ let us start oxidation of fatty acid. This is the first fate of fatty acid. It is oxidation and generation of energy. It is the first fate. The amount of energy obtained from this fatty acid oxidation is higher than glucose oxidation, because fatty acid, they are found in highly reduced form, or they are found in low oxidation state. As a result, whenever they are oxidized, they will give us more number of ATP or energy than carbohydrate due to this low oxidation state. This is very important. You have to compare the amount of energy between different nutrients. Nutrients like glucose, fatty acid, and protein or amino acid, they will give us energy upon their oxidation, but different amount of energy is obtained due to this different number of electron and hydrogen present in this nutrient will give us a different amount of energy. So more number of ATP is obtained from this fatty acid oxidation. It is the first phase. So provide energy for the cellular and metabolic activities in our body. Daily recommended allowance for the fat in our diet is it is found between 29 to 31%. This is for American standard dietary food intake. For Ethiopian, still it is not known, or it is not studied. Based on this American daily recommended daily allowance, it is recommended to intake 29 to 31% of fats in our food. It is recommended. Okay, the Ethiopian does it. It is not studied, or it is not calculated. Through beta oxidation, there are different oxidation in fatty acid. The primary or the default mechanism of fatty acid oxidation is through beta oxidation. Beta indicate the carbon location. As you know, the carbons, they are labeled either number or Greek letter alpha, beta. The other, it is by omega system, they are labeled. So number, it is started from the functional group, carboxylic acid functional group. So this is the one structure of fatty acid with hydrocarbon chain and carboxylic acid functional group. The functional group is our carbon number 1, and the next carbon is labeled as carbon number 2, carbon number 3. It will be continue like this. If you are using a Greek letter, alpha is given, alpha, the first letter, it is given for carbon which is found next to this carboxylic acid. Carboxylic acid, it is a functional group. And then next to this carbon, we give alpha. So alpha, beta, gamma, delta, it will be given sequentially up to the end. So the end of fatty acid, it is omega, and the first carbon is an alpha. Oxidation, either it is occur between alpha and beta. If this oxidation is found between alpha and beta carbon, we call it beta oxidation. Alpha oxidation, the second type, it is alpha oxidation. Alpha oxidation is occur between the functional group carboxylic acid and the first or the alpha carbon, or carbon number 1 and carbon number 2. Between the two, oxidation it is occur during alpha oxidation. The third type of fatty acid oxidation, it is called omega oxidation. It is occur from the end of fatty acid. From the end of fatty acid, there will be oxidation. So there are alpha oxidation, beta oxidation, and omega oxidation. Which one is more frequently occur, or it is default mechanism of fatty acid oxidation? It is beta oxidation. Alpha and omega, they are minor, plus they are unique for some type of fatty acid oxidation. Some type of fatty acid, they may use omega oxidation or an alpha oxidation based on their structure. Okay, now let us see the beta oxidation. Beta oxidation, it have 3 stages or phase. There are 3 phases. By the first phase, there is oxidation of fatty acid, beta oxidation, chopping fatty acid by 2 carbon. At a time, 2 carbons are cleaved from this fatty acid, starting from the first carbon. Carbon number 1 and carbon number 2 are cleaved first, and then 3 and 4, they are cleaved on the second round. This will be continue until the end of fatty acid oxidation. So this is the first phase. By the second phase, those cleaved carbons or acetyl-CoA, which is formed by this beta oxidation, join a Krebs cycle or citric acid cycle, will be occur by the second stage or phase. Through this cycle, electrons, hydrogen atom will be transferred to coenzyme. Coenzymes are formed, reduced coenzymes are formed. The carbon, they are released as carbon dioxide carbons. They are released as a carbon dioxide. Electrons and hydrogen atom will be transferred to coenzyme. Coenzyme carry those electron and hydrogen atom from acetyl-CoA. The reduced coenzyme now join or enter into the electron transport chain. Through electron transport chain or respiratory chain, they will be oxidized, re-oxidized in the electron transport chain. They transfer electron and hydrogen atom to this electron transport chain. As a result, we will have ATP formation. By ATP synthase channel, there is ATP formation. Yesterday, you have learned this part, electron transport chain. Before yesterday class, you have learned the Krebs cycle or TCA cycle during carbohydrate metabolism. Today, you are going to see the beta oxidation, how fatty acid is chopped or cleaved by 2 carbons will be discussed. Oxidation, as you know, oxidation is removal of hydrogen atom or loss of electron or addition of oxygen makes the molecule oxidized. Reduction is opposites. That means gain of electron, addition of hydrogen atom, or removal of oxygen makes the molecule reduced. So there is oxidation. By this oxidation, we'll have a reduced coenzyme formation. There is oxidation between carbon number 2 and 3 here. Carbon number 2 and 3, between these 2 carbon, there is oxidation. Through this oxidation, we'll have removal of hydrogen atom and loss of electron. Those electron and hydrogen atom will be accepted by a coenzyme. Coenzymes are formed here in the beta oxidation plus in the Krebs cycle. So Krebs cycle and the electron transport chain will be omitted or escaped by this discussion. We are only focusing on the stage 1, that is beta oxidation. There are 4 sequential reaction to cleave a 2 carbon, to break down the 2 carbon or to cleave the 2 carbon. There are 4 sequential reactions. First oxidation, the second hydration, the third reaction is the second oxidation, plus finally the fourth reaction is cleavage. Cleaving the 2 carbons are occur. Okay. The second fate of fatty acid, it is for providing energy for the brain during starvation. How it provides energy for the brain? It is by making ketone bodies. Ketone bodies are formed, and as a result, we'll have energy source to the brain. As you know, fatty acid, they cannot cross. They are not able to cross the blood-brain barrier. So we have to convert fatty acid or oxidize fatty acid to acetyl CoA. Acetyl CoA will be used for ketone body synthesis. The ketone body will be used for as energy source in the brain. The second one is gluconeogenesis will be occur. The gluconeogenesis, it is occur from propionyl CoA. Propionyl CoA, we'll see later how it is formed. It is formed from odd carbon number fatty acid. Odd carbon number fatty acid. Usually fatty acid, they do have even number, but sometimes they may have odd number carbon fatty acid. Whenever we cleave 2 carbon from this fatty acid, the remaining 3 carbon called propionyl CoA will be converted to succinyl CoA. The succinyl CoA will be converted to oxaloacetate, then oxaloacetate finally converted into glucose through gluconeogenesis. So there is gluconeogenesis number 1 from propionyl CoA. Number 2, acetyl CoA will be converted to ketone body. Ketone body will be used as a source of energy to the brain. This is during starvation. We'll have as a fuel or provide fuel to the brain, to the our brain will have this energy production. We'll see types of ketone body, how it is formed, the reaction, how it's occur, plus the organ location will be discussed later for this ketone body synthesis. Okay. The second one is the third fate of fatty acid. It is for giving heat or generation of heats for hibernating animals. Hibernating animal, they do have high lipid content, or there is subcutaneous tissue lipid that will be oxidized for the formation of heat or generation of heat. It is occur in the hibernating animals. There are 2 types of adipose tissue. One of adipose tissue, it will be used for storage of lipids or fats in the white adipose tissue. Brown one, the brown adipose tissue, it will be used for thermogenesis, for heat generation for those hibernating animals. How thermogenesis is occur? I hope yesterday you have learned in the electron transport chain. The one which is accumulated, the proton which is accumulated between the inner and outer membrane of mitochondria will be leaked into matrix without ATP from ATPase channel. Normally, this proton will return back to the matrix through ATPase channel, but during thermogenesis, protons will be returned or leaked into matrix rather than through ATP synthase channel. Okay, ATP synthase channel. Whenever we have ATP formation, there is returning protons through this ATP synthase channel. Whenever there is thermogenesis or uncoupler, there is leakage of proton to the matrix without through the ATP synthase channel. In that case, we'll have or we lost the energy in the form of heat. Heat generation will be occur upon returning this proton to matrix rather than this ATP synthase channel. So we don't have ATP formation. There is no ATP formation because the hydrogen atom or the proton it is not returned back to matrix through ATP synthase channel. It is leaked into matrix. As a result, we'll have in a heat formation, or the energy found in the electron or proton, it will be lost as heat. So we have thermogenesis, or heat generation will be occur upon this fatty acid oxidation. This is the third fate of fatty acid in our body. This one, it is a protein molecule, or thermogenin. It is a protein or uncoupler protein in the inner membrane of mitochondria, found in the brown adipose tissue, and generate heat rather than ATP formation from this electron transport chain. This was discussed yesterday by Tensai. Okay, the fourth fate of fatty acid, or functions of fatty acid in our body is it is to synthesize intermediate molecule. Fatty acid oxidation generates intermediate molecule in the synthetic process. For example, phospholipid in the membrane. It is important to make eicosanoids. Eicosanoid, again, it is important to make prostaglandin. It is allergy mediator. Prostaglandin mediate allergy or inflammation mediated by this prostaglandin. Leukotrienes, thromboxane, they are formed by eicosanoids. Eicosanoid, or arachidonic acid. Arachidonic acid, it is a 20 carbon fatty acid, usually found in the membrane lipid. So this will be used for the formation of prostaglandin and leukotriene thromboxane, and to have allergy mediation or inflammatory mediator molecules will be formed by these reactions. Acetyl-CoA, this will be used for cholesterol synthesis. You see, the cholesterol synthesis or ketone body synthesis will be occurred by this acetyl-CoA, which is formed from fatty acid oxidation. Propionyl-CoA, again, it will be used for making glucose through gluconeogenesis. So this intermediate molecule will be used for synthesis of different molecule. They will be used for making different molecule from those intermediate found in fatty acid oxidation. Which tissues are active on fatty acid oxidation? Those tissues, they have mitochondria first. They need to have mitochondria plus enough amount of oxygen. It is required to have this fatty acid oxidation. To be able to oxidize fatty acid, they need to have mitochondria, and plenty of oxygen is required. Number one. Number two, in the brain, fatty acid, it cannot cross. Brain, it have mitochondria, but fatty acid cannot cross the blood-brain barrier. As a result, fatty acid oxidation does not occur in the brain cell. So except RBC and brain, other cell, they can oxidize fatty acid to generate energy or ATP formation. It will be occurred by the other type of cell. Why RBC cells don't have this beta oxidation or fatty acid oxidation? Because the mitochondria is absent. It is devoid of mitochondria in the RBC cell. Brain cell, in the brain cell, fatty acid cannot enter into the brain because of this barrier. Fatty acid oxidation, it is most active in tissues which are active, metabolically active tissues. They use fatty acid as a source of energy. These are skeletal and heart muscle. They have high energy demand. As a result, they use fatty acid as a source of energy in addition to glucose. All cells, they prefer to use glucose as a source of energy, but those who need high amount of energy, they use the secondary energy preference called fatty acid will be oxidized in those tissue types. So normally 60 to 90% of the energy required for the heart muscle contraction is derived from this fatty acid oxidation. Um, fatty acid oxidation increased in the fasted states during glucose is depleted or glycogen is depleted. There is fatty acid oxidation. I have told you before, our primary source of energy it is glucose, but whenever it is deficient, tissues will start to utilize fatty acid as a source of energy. So it is activated in the fasting period. In the fasting period, we will have low carbohydrate intake or glycogen formation in the liver. In that case, fatty acid oxidation will be stimulated. During exercise, again, there is a demand of high energy by the muscle. In that case, fatty acid oxidation will be increased. So there are two conditions. Number one, in the fasting period and during exercise, fatty acid oxidation will be increased. Fatty acids, after they are released from triacylglycerol, they'll be taken by the cell. They'll be transported into target cell. Assume it is a muscle cell. So they will be taken into muscle cell by using different transporter. There are membrane transporter or carrier molecule which can transport fatty acid into the cell. The first one is fatty acyl transporter or CD36. It is a membrane protein transporter for fatty acid. Passively or without energy consumption, fatty acid it will be taken into the cell by using this transporter. In the heart muscle, skeletal muscle, adipocyte, intestine, they do have this transporter. And then as a result, they can take fatty acid from the blood to the cell. This is an abbreviation, fatty acyl translocase enzyme, found in the membrane, plasma membrane, transports fatty acid into cytoplasm of the cell. After it is transported, fatty acid it will be activated. There will be activation of fatty acid into active form. Okay, fatty acid activation. Here we have addition of CoA, coenzyme A. It is added to fatty acid for the formation of activated fatty acid, fatty acyl CoA or acyl CoA. It is formed. This is similar, like enterocyte fatty acid activation. It is similar. The enzyme also it is similar. Fatty acyl CoA synthetase enzyme catalyze this reaction with ATP consumption. The ATP is cleaved or hydrolyzed into AMP. Because of this, we said it is equivalent with 2 ATP. Actually, it is 1 ATP consumption, but we use two high energy phosphate bond. Okay, two high energy phosphate bonds are used for the activation of fatty acid into fatty acyl CoA. Therefore, during activation of fatty acid, we consume 2 ATP. 2 ATPs are consumed for the activation of fatty acid. After it is activated, it will be translocated into mitochondria. So this will be taken into mitochondria to have beta oxidation. This is again how it is activated. It is explained by this. Once the fatty acid is taken into the cell or inside the cell, fatty acid must be activated before they can be metabolized. It is catalyzed by acyl CoA synthetase, localized to 3 different sites in the cell. There are 3 different sites. Cytosolic phase of endoplasmic reticulum, outer mitochondrial membrane, and peroxisomal membrane. They contain, they do have this enzyme and activate the fatty acid. Here, the endoplasmic reticulum, it is very important, or to have this enzyme because the endogenous synthesis of fatty acid, it is located, or modification of fatty acid, it is occur in the endoplasmic reticulum. Mitochondrial membrane to have this enzyme because beta oxidation or oxidation of fatty acid it is occur in the mitochondria organelle. Plus peroxisome, here the very long chain fatty acids which to have carbon number greater than 20 22 greater than 22 carbon containing fatty acid, they will be shortened first in the peroxisome. So we have beta oxidation in the peroxisome also. Therefore, we have this enzyme at peroxisomal membrane. So this parts of the cell or site of the cell to have this enzyme and activate fatty acid into active form. There are three isoenzyme isoform for those fatty acid which to have different carbon lengths. Short chain fatty acid, they need short chain fatty acid CoA synthetase. Medium chain fatty acid, they do have their own enzyme isoform called medium chain fatty acid CoA synthetase. Long chain fatty acid, they do have their own enzyme called long chain fatty acid CoA synthetase enzyme. Okay, isoforms are for different fatty acid lengths when they use a different fatty acid substrate that is based on different number of carbon in the fatty acid. Fatty acyl CoA, this is the fate of fatty acyl CoA. Once it is activated, it will be oxidized and generate energy, or there will be ketogenesis from this acetyl CoA. There will be storage in the white adipose tissue in the form of triacylglycerol, or this will be incorporated into phospholipid and sphingolipid and found in the membrane lipid, or will be used for making the cell membrane, or cell membrane will be formed by making phospholipid and the sphingophospholipid. Okay, these are the three fates of fatty acyl CoA. Now we are looking the beta oxidation. Beta oxidation, it is occur after it is activated by fatty acyl CoA synthetase. There are different isoform for those different carbon lengths. The short and medium, short chain fatty acyl CoA synthetase, medium chain fatty acyl CoA synthetase, long chain fatty acyl CoA synthetase. These are different isoforms of fatty acyl CoA synthetase. Types of fatty acid oxidation, there are three types: alpha oxidation, beta oxidation, and omega oxidation. The major one is beta oxidation, but some fatty acid do have alpha oxidation and omega oxidation. Whenever, for example, branched structure is found in the fatty acid, we'll have alpha oxidation. Whenever they start from the end of fatty acid, there will be omega oxidation, but the second one is the one which is default in our body. Transport of long chain fatty acid into mitochondria, how they are transported into mitochondria, because coenzyme A added to this fatty acid is impermeable. The one which is added to fatty acid, this is how it is activated. Once it is activated, we have this fatty acyl CoA thioether group. It is added to this fatty acid. This one, it is impermeable through mitochondrial membrane, through inner and outer membrane of mitochondria. It is impermeable. We have to replace this one. We have to replace the coenzyme A. How it is replaced? Which molecule replaced the coenzyme? It is replaced by carnitine. Carnitine, it is a peptide molecule, peptide made from lysine and methionine amino acid. Lysine and methionine, they form carnitine, either endogenously synthesized or obtained from dietary food, especially from meat. Meat consumption, it is very important to have this carnitine peptide. Therefore, this one is very important to transport fatty acid into mitochondria. This will replace coenzyme A. As a result, fatty acid will be transported into mitochondria to have beta oxidation. Beta oxidation, as you know, it is located or found in mitochondrial matrix. The enzymes, they are found in the mitochondrial matrix. Inner mitochondrial membrane, it is impermeable to coenzyme A. We need a specialized carrier which can transport the long chain fatty acyl group from cytoplasm to mitochondria. This carrier called carnitine. So carnitine derivative transports the acyl group into mitochondria. Once it is transported, again carnitine will be removed or will be replaced by coenzyme A. So coenzyme A, it is re-added or replace carnitine and form fatty acyl CoA in the matrix of mitochondria. So this carnitine shuttle, it is a rate limiting step for beta oxidation. Beta oxidation is controlled by this carnitine shuttle, or the enzyme which are involved in the replacement of coenzyme A by carnitine and carnitine removal and addition of carnitine. It is catalyzed by enzymes. Those enzymes, they are controlled by our hormones at gene level or substrate level. They are controlled. So this is the level where beta oxidation is regulated or controlled by limiting transport of fatty acid into mitochondria. If it is not transported, we don't have beta oxidation. If it is transported, then beta oxidation will be occur. Fatty acyl carnitine is formed. Carnitine, it is look like this. It have this kind of structure. It will be added to fatty acid and then form fatty acyl carnitine. This will be transported into mitochondria to have a beta oxidation. First, by carnitine acyl transferase 1, this is the enzyme. Carnitine acyl transferase 1 replace coenzyme A by carnitine. This will be taken off. Coenzyme A will be taken off and add carnitine to acyl group. So we have fatty acyl carnitine formation by this enzyme. And then after crossing the inner mitochondrial membrane, the second enzyme remove carnitine and add coenzyme A. This is a simple diagram showing how it is activated plus transport into mitochondria. It is represented by this diagram. Acyl CoA synthetase, this one is acyl CoA synthetase enzyme, activate fatty acid. Carnitine acyl transferase 1 replace coenzyme A by carnitine, then transport acyl group into the matrix. Now let's see. Let's take one fatty acid, palmitate, which is 16 carbon fatty acid, 16 carbon 16 carbon yellow fatty acid. Now this will be activated into palmitoyl CoA by acyl CoA synthetase enzyme. Consume 1 ATP. This ATP is hydrolyzed to AMP. Don't forget, for one fatty acid activation, we consume two ATP equivalents. 2 ATP equivalents are consumed. Now this is activated. Once it is activated, coenzyme A replaced by carnitine, which is catalyzed by this enzyme. Carnitine acyl transferase 1 replace coenzyme A by carnitine. So we have this. Carnitine is added to this fatty acid. Palmitoyl carnitine is formed, and coenzyme A will be taken off. This is this one. This is the outer membrane of mitochondria. This is outer membrane. As you know, membrane, it is a double layer membrane, outer leaflet and the inner leaflet of the outer membrane of mitochondria. The inner membrane, okay, this is intermembrane space. It is the space between the two membrane of mitochondria. Okay, here we have one notice. The defect in carnitine acyl transferase 1 or carnitine palmitoyl transferase 1 defect in the muscle cause severe muscle weakness because of unable to use fatty acid as a source of energy, unable to use fatty acid in the muscle. As a result, we have failure to generate energy from this fatty acid oxidation during exercise. So this is very important. Regulation of carnitine acyl transferase 1. This one, it is regulated by malonyl CoA. Malonyl CoA, it is an intermediate molecule formed during fatty acid synthesis. Malonyl CoA formed during fatty acid synthesis. Synthesis of fatty acid will form malonyl CoA. Then this will inhibit transport of fatty acid into mitochondria to have beta oxidation. ምንድን ነው የሚለው ፋቲ አሲድ ኦክሲዴሽን ፕላስ fatty acid synthesis, they are not occur at the same time. They are not occur simultaneously. Whenever we have synthesis, we have to inhibit oxidation. Whenever we have oxidation, then we have to inhibit synthesis. They are not occur simultaneously. So if our cell is under synthesis, there is malonyl CoA. Malonyl CoA inhibit this enzyme. And then when this enzyme is inhibited, beta oxidation will be inhibited. If we are on the degradation of fatty acid or oxidation of fatty acid, and then synthesis will be inhibited by another mechanism. There are other mechanism of regulation of synthesis. We'll see later. So this enzyme, it is our rate limiting enzyme. It is controlled or regulated by this malonyl CoA allosterically. Allosteric inhibition of this enzyme, it is occurred by malonyl CoA. This malonyl CoA concentration will be high during fatty acid synthesis. During synthesis, this malonyl CoA amount will be increased, then inhibit carnitine acyl transferase 1. As a result, beta oxidation will be inhibited. This is very important. Whenever this one is found, then this will prevent simultaneous synthesis and degradation of fatty acid. On the second membrane, or on the inner mitochondrial membrane, we re-add coenzyme 8 on acyl group. We have the second enzyme, carnitine acyl transferase 2. Carnitine acyl transferase 2 replace or add coenzyme A by removing carnitine. Remove carnitine, add coenzyme A on acyl group. This one is occur on the inner membrane of mitochondria. The outer membrane, inner membrane space, intermembrane space, plus the inner membrane of mitochondria. On the outer membrane, we add carnitine. By the inner membrane of mitochondria, we replace or we remove carnitine and add carnitine. Mitochondrial uptake via palmitoyl carnitine. Okay, carnitine acyl carnitine translocase enzyme. This one translocase enzyme. This one exchange fatty acyl carnitine into matrix and exports or translocates carnitine out of mitochondria. We'll see later. Okay, here this translocase enzyme, it is important to exchange palmitoyl CoA, palmitoyl carnitine into matrix and carnitine out from this matrix to cytoplasm. So we will have this exchange mechanism using this translocase enzyme. Translocase enzyme finally makes us to recycle this one. Recycling carnitine, it is occurred by using this translocase enzyme. Finally, we found this fatty acid or activated fatty acid in the cytoplasm of the cell. So this one, it can undergo through this beta oxidation because it is found in the matrix of mitochondria. Again, this one is discussed by this diagram. We have two enzyme, carnitine acyl transferase 1 and 2. They will transport or replace coenzyme A by carnitine, then carnitine by coenzyme A. They will be help during this transport of fatty acid into the matrix. Translocase enzyme, it is important to exchange fatty acyl carnitine into the matrix and carnitine out of the matrix. It will be exchanged using this translocase enzyme. Okay, this is the outer membrane of mitochondria, the inner membrane of mitochondria. By the outer membrane of mitochondria, there will be replacement of coenzyme A. Carnitine is added, coenzyme A is removed, and then we have this carnitine, fatty acyl carnitine is formed. Once this one is formed, again, it will be taken into the cell, into the matrix. It will be transported by translocase enzyme. Once it is in the matrix, the second enzyme, carnitine acyl transferase 2, will remove carnitine and add coenzyme A. Carnitine will be removed, coenzyme A will be added. So carnitine returned back to the into the inner membrane space of mitochondria to have the second transport of fatty acid. So this will be frequently occur as much as we have this fatty acid in the inner membrane of mitochondria. So this is how it is transported into the matrix. Um, source of carnitine, I told you before, this one is made by two amino acids, lysine and methionine amino acid form carnitine. The synthesis, it is occur in the liver and kidney. Liver and kidney, they are organ location where carnitine is formed. So the skeletal and heart muscle, they cannot synthesize carnitine. Always they are dependent on diets or endogenously synthesized carnitine. On the diets, what are the source of carnitine? The meat contain high amount of carnitine. It will be the source of this carnitine for skeletal muscle and heart muscle. Methionine, as you know from your module 1 class, it do have sulfur. It is one of sulfur containing amino acid. It is important to make S-adenosine methionine SAM. By this we can donate one carbon, or methyl group will be donated one carbon. CH3 group will be donated to different chemical reactions. For example, vitamin B12 synthesis, it is occur with the help of S-adenosine methionine. S-adenosine methionine, it is very important to donate one carbon molecule to the chemical reactions. And plus vitamin C ascorbic acid is also required for this reaction. So S-adenosine methionine, it is formed by methionine. Methionine, it is important to make this part. Lysine and methionine, again, they make carnitine. Vitamins also is important to have this reaction. If you have carnitine deficiency, as a result, we cannot transport fatty acid into the matrix, and then we fail to use fatty acid as a source of energy in our body. That may be due to carnitine acyltransferase 1 deficiency, genetic defects, or genetic deficiency of this enzyme may occur. As a result, we'll have hypoglycemia, coma, or death occur. Carnitine acyltransferase 2 deficiency, this one mainly affects skeletal and cardiac muscle. Again, this is genetic defects. Both are enzymes. Enzymes are, they are proteins. Proteins, they are made by the help of DNA information. DNA dictates amino acid sequence in the protein. Whenever there is defect in the gene or DNA, as a result, we'll have problem in the proteins function, like enzyme will be defective. Defect in renal tubular reabsorption of carnitine. Carnitine is important, so we have to reabsorb carnitine. As a result, we can use fatty acid as a source of energy. If there is a defect in kidney, and then we are not able to reabsorb carnitine into the body, as a result, we'll have deficiency of carnitine. This will finally leads to energy failure. Defect in carnitine uptake by the cell. This is another causes of carnitine deficiency in our body. Secondary causes, liver disease, because it is organ location where it is synthesized. Malnutrition, whenever there is strict vegetarian. Water dietary source or food source of carnitine, they are meats. So if you are strict vegetarian, then we are not able to get carnitine from diet. Increased metabolic demand, in that case, the available carnitine will not be sufficient to the transport fatty acid into the matrix. Hemodialysis will remove carnitine as a result of this dialysis process. The short and medium chain fatty acids, they don't require carnitine. Again, this is the second exceptional point on short and medium chain fatty acid. Previously, we have seen the short and medium chain fatty acid, they are absorbed directly to portal blood vessel. Again, here, during their transport into mitochondria, they don't require carnitine. Carnitine is not required by the short and medium chain fatty acids. Directly, they can be taken into the cell. They are activated into their CoA form inside the mitochondrial matrix. Okay. not they are not inhibited even by this malonyl CoA because we don't need carnitine for their transport. Reactions of beta oxidation. There are 4 sequential reactions to cleave a 2 carbon. To cleave 2 carbon, there are 4 sequential reactions. Don't forget, we have 4 reactions. Let's see the sequential reactions. በነገራችን ላይ እዚህ ጋር ማስታወስ ያለባችሁ it is an oxidation process. Oxidation ማለት removal of hydrogen, loss of electron, and addition of oxygen. By these 3, either of this process make the reaction is oxidized, or the molecule is oxidized. There is oxidation between carbon number 2 and 3. Upon this oxidation, we lose electron, remove hydrogen atom. Those electron and hydrogen atom will be taken or accepted by those coenzyme. FAD, flavin adenine dinucleotide, NADH, nicotinamide adenine dinucleotide, accept the lost electron and hydrogen atom. The 2 carbon which are cleaved by this beta oxidation are released as acetyl-CoA. We have acetyl-CoA formation per 1 cycle of beta oxidation. This is 1 cycle of beta oxidation, and then the second cycle, the third cycle, up to the end of fatty acid oxidation, we'll have this cycle of beta oxidation. There are 4 steps for 1 round of beta oxidation. For 1 round of beta oxidation, there are 4 enzymatic reactions. The first reaction is dehydrogenation, removal of hydrogen atom, loss of electron between the second and the third carbon. Between the second and the third carbon of fatty acid, there is oxidation. This one is catalyzed by acyl-CoA dehydrogenase enzyme. Acyl-CoA dehydrogenase enzyme catalyze the first oxidation. The second reaction, the second enzymatic reaction is hydration. Hydration means addition of water. We add water to the double bond, and it is catalyzed by enoyl-CoA hydratase enzyme catalyze this reaction. The third reaction is the second oxidation. The second dehydrogenation reaction, it is occur. This one is linked with NAD. The first one is linked with FAD, flavin adenine dinucleotide, linked with this reaction. Those electron and hydrogen atom will be accepted by this coenzyme. On the second oxidation, electron and hydrogen atom will be accepted by this coenzyme NAD. Nicotinamide adenine dinucleotide will accept the electron. So we form the enzyme. It is catalyzed by 3-hydroxyl acyl-CoA dehydrogenase enzyme catalyze this reaction. The fourth reaction, theolytic cleavage, cutting the 2 carbon. It is occurred by beta ketoacyl-CoA thiolase enzyme. In short, we call it thiolase. This one is a thiolase enzyme. The first one is dehydrogenase enzyme, hydratase enzyme, dehydrogenase enzyme, and thiolase enzyme. They catalyze this oxidation process. So this is per 1 round. So every round, on every round, there are 4 reactions. At the end of these 4 reactions, we cleave 2 carbon. By the first oxidation, there is 1 coenzyme formation. FADH2 will be formed. By the second oxidation, there is NADH formation. By the third reaction, there is the release of acetyl-CoA. Acetyl-CoA will be cleaved. Okay, now let us see the reactions step by step. And one እናያለን. Okay, this is the activated fatty acid. Activated fatty acid. This is our coenzyme A. This is our carboxyl group. So next to this carboxyl group, we have carbon number 2, carbon number 3. There is oxidation between carbon number 2 and carbon number 3. Carbon number 2 and carbon number 3. They are the carbon location where oxidation is occur. Okay, by each carbon, by 2 of the 2 carbon, carbon number 2 and carbon number 3, they lost electron and hydrogen atom. They lost hydrogen. One hydrogen will be lost. Together with hydrogen, electron will be again lost. So we have a double bond formation after this oxidation. The single bond will be converted into double bond. By this double bond, you can see one geometrical isomerism. The geometrical isomerism that is called trans isomer will be formed after the first oxidation. Naturally, fatty acid do have a cis double bond, but during beta oxidation, we form a trans double bond. So this one is the one formed during beta oxidation. Trans double bond will be formed by the first oxidation. Trans. The hydrogen atom next to this double bond will be found in opposite direction. They are found in opposite direction. We call it a trans. But in the natural fatty acid structure, we found double bond in the same direction, or in the cis configuration. Okay. So it is another ማለት ነው። እሺ oxidation of fatty acid to trans double bond in oil-CoA by acyl-CoA dehydrogenase enzyme. During this reaction, we will form one reduced coenzyme called FADH2 will be formed. The second reaction is addition of water. Water will be added. The OH group of water will be added to carbon number 3. The hydrogen atom of water will be given to this carbon number 2. So the double bond finally converted to single bond. Okay, we add one alcohol functional group to this carbon number 3, and it is catalyzed by enoyl-CoA hydratase enzyme catalyze this water addition. The trans enoyl-CoA finally converted to L3 hydroxyl acyl CoA will be formed. The third reaction is the second oxidation into 3 keto acyl CoA. This will be oxidized. Carbon number 3 only will be oxidized. This carbon will be oxidized. The 2 hydrogen atom will be removed, and hydrogen atom electron will be lost. Okay, when this carbon is oxidized, we have this carboxyl group formation. This carboxyl group, it is the same end with the first carboxyl group. Okay, so we make the same ending. After we have this, we can have cleavage. This bond can be cleaved with the addition of another coenzyme A. We add another coenzyme A, and as a result, we will have this oxidation. So by this oxidation, there is one NADH formation. One coenzyme will be formed during this oxidation process. The fourth reaction, theolysis or cleavage of acetyl-CoA from the end of fatty acid, it is occur by the addition of coenzyme A. So we have a similar ending. Similar ending will be occur after this theolytic cleavage. Theolytic cleavage gives us this the same ending. This one we call it a 2-carbon shortened fatty acid. 2-carbon shortened fatty acid or acyl-CoA will be formed, and acetyl-CoA or 2-carbon molecule will be formed or released from this fatty acid at the end of these 4 reactions. So this reaction, these 4 reactions will be occur at every round of beta oxidation. At every round, we'll have one FADH2 formation, one NADH, and acetyl-CoA will be formed. So this one repeated for another cycle. If you have 16 carbon fatty acid, this will be occur 7 times. Beta oxidation will be 7 times will be occur. 18 carbon fatty acid to have 8 times. 18 carbon, it is 7 times. Okay. 16 carbon, it is 6 times beta oxidation will be occur. So the end product of 16 carbon fatty acid oxidation, it is called it is acetyl-CoA, NADH, and FADH2 will be formed. Again, this one is discussed. Let's see the end product formation. When products of 16 carbon fatty acid oxidation. This one, acyl-CoA dehydrogenase enzyme, it have 3 isoform based on the length of fatty acid again. For long chain fatty acid, they have a long chain acyl-CoA dehydrogenase enzyme. Long chain fatty acid to have carbon number greater than 12. Medium chain fatty acids, they have their own acyl-CoA dehydrogenase enzyme called medium chain acyl-CoA dehydrogenase enzyme. This medium chain fatty acid to have carbon number between 6 to 12. Short chain fatty acid to have carbon number between 4 to 6. 4 to 6 carbon, it is found in the short chain fatty acid. It is catalyzed, it is oxidized by acyl-CoA dehydrogenase enzyme. The medium chain fatty acid acyl-CoA dehydrogenase enzyme deficiency is again commonly occur in the inborn error of metabolism, that is due to genetic defects. We'll have this enzyme deficiency, and then we will not able to use the medium chain fatty acid as a source of energy. Medium chain fatty acids usually they are formed from milk fats in the neonates. So whenever there is an inborn error in this enzyme isoform, those children or neonates, they cannot use fatty acid as a source of energy from milk. Milk consumption will be avoided. Fate of acetyl-CoA, it can be oxidized through Krebs cycle and electron transport chain. In the liver organ only, this acetyl-CoA will be used for ketone body synthesis or ketogenesis will be occur in the liver organ. The fate of FADH2 and NADH, they will be re-oxidized in the electron transport chain. As a result, they will give us ATP, produce ATP upon their oxidation in the electron transport chain. This is Krebs cycle. This is a Krebs cycle. Acetyl-CoA condensed with oxaloacetate for the formation of citrate. It is catalyzed by citrate synthase enzyme. The second reaction is aconitase enzyme catalyzed the isoform isomerization of citrate to isocitrate. The first oxidation it is occur and convert isocitrate to alpha ketoglutarate. This one is catalyzed by isocitrate dehydrogenase enzyme catalyze this reaction. It is the first oxidation process, and one of the two carbon in the acetyl-CoA are removed from this oxidation process. Again, the next step, it is the second oxidation. We have the second oxidation. Alpha ketoglutaric acid will be oxidized to succinyl-CoA. Again, the second carbon here will be released in the form of carbon dioxide. So what is left? The left is electron and hydrogen atom. The electron, some of the electron is transferred here to the NAD. NADH is formed, and again NADH it is formed here. Substrate level phosphorylation, again it is occur here. Substrate level phosphorylation, as you know, substrate level phosphorylation is occur by high energy bond breakdown. High energy substrate breakdown will release energy. That energy will be used for the phosphorylation of GDP to GTP. GDP accepts phosphate or add phosphate with the help of high energy bond breakdown in the succinyl-CoA. Succinate will be oxidized into fumarate by succinate dehydrogenase enzyme. Here FAD will be reduced to FADH2. Hydratase or fumarase enzyme add water for the formation of malate. Malate again oxidized to oxaloacetate. So we have transferring electron and hydrogen atom to the coenzyme. Coenzymes are formed here. Per one acetyl-CoA, there will be 3 NADH, 1, 2, 3 NADH are formed, plus 1 GTP by substrate level phosphorylation, and 1 FADH2 will be formed per 1 acetyl-CoA. This is the amount of the number of coenzyme and substrate level phosphorylation. If you have 16 carbon fatty acid, how many NADH, how many FADH2, and how many GTPs are formed, and the total number of ATP per 16 carbon fatty acid we will discuss. This is the electron transport chain. We will not discuss this one because you have learned it from yesterday's class. The total ATP formation from the oxidation of 1 mole of palmitic acid, 16 carbon fatty acid, 2 carbon dioxide, and water. Let's calculate the number of acetyl-CoA, the number of NADH and FADH2 formation by the 16 carbon fatty acid oxidation. There are 7 cycles. 7 cycles of beta oxidation are occur. Per each cycle, there is NADH and FADH2 formation, and 7 acetyl-CoA will be formed. But at the end, we'll have 2 carbon molecule. 1 acetyl-CoA will be formed as a leftover. Together they form 8 acetyl-CoA will be formed. So per 1 acetyl-CoA, you know the number of NADH, FADH2, and GTP. Per 1 NADH, there is 3 ATP formation. Per 1 FADH2, there is 2 ATP formation. Then we can calculate the amount of ATP formation per 16 carbon fatty acid, or per 1 molecule of palmitoyl CoA, how many ATP will be generated. Now it can be calculated. But during fatty acid activation, we consume 2 ATP. For palmitoyl activation, 2 ATPs are consumed. We will subtract these two ATP from the total ATP generation from this palmitoyl CoA oxidation. Okay. Again, this is a repetition of the beta oxidation. Palmitoyl CoA, it is oxidized by this beta oxidation in repetitively in different beta oxidation cycle. Per each cycle, there is 4 reactions. There are 4 reactions. First oxidation, hydration, the second oxidation, and thiolysis. This is one cycle. This will be repeated 6 times for 18 carbon fatty acid. If you have 18 18 carbon ከሆነ ደግሞ number of cycle ይጨምራል። 20 ሲሆን ልክ እንደዚሁ ሳይክሉ ይጨምራል። ሳይክሉ በጨመረ ቁጥር የምናገኘው ውጤትም ይጨምራል። እሺ acetyl CoA join the citric acid cycle and produce or transfer its electron to coenzyme. FADH2 and NADH will join the electron transport chain. As a result, we will have ATP formation. ለምሳሌ FADH2 will give us 2 ATP upon through this respiratory chain. NADH will give us 3 ATP in the electron transport chain. Acetyl CoA, one acetyl CoA will give us 12 ATP. 12 ATP በቀላሉ ይሄን ካልኩሌት ማድረግ እንችላለን። አንድ አሲታይል CoA 3 ናዴች ነው የሚሰጠን። አንድ FADH2 እና አንድ GTP ይሰጠናል። ካልኩሌት ስናደርግ ስንደምራቸው 12 ይመጣል። So 8 acetyl CoA times 12 it is 96. 7 NADH times 3 it is 21. 7 FADH2 times 2 it is 14. Together we have 131 ATPs are formed. Minus 2 ATP. 2 ATPs are utilized during formation of acyl CoA. The net ATP yield is 129 ATP will be generated upon 16 carbon fatty acid oxidation. So this is it is very excess, or it is too much than glucose oxidation. If glucose is completely oxidized, if one glucose molecule is completely oxidized, we'll have 38 ATP formation. 38 ATP. Per this 116 carbon fatty acid oxidation, we have 129 ATP. This one is due to the number of hydrogen and electron in the fatty acid. Fatty acid, they are highly reduced form, or they are found in low oxidation state. Upon their oxidation, they will generate high amount of energy than glucose molecule. Oxidation of unsaturated fatty acid. እሺ
ሄሎ
አቤት ናት።
ሄሎ ሰዓቱ ደርሷል። ዛሬም ከታክላት ላቦራቶሪ ስላለ ባስም ስለሚያመልጠን ማለት አንደርስታንድ ሄዱ።
ስለዚህ ነገ ክላስ ይኖራል አይደል?
ነገ ክላስ አለ።
We will start tomorrow by beta oxidation of unsaturated fatty acid, odd carbon number fatty acid oxidation ነገ እንጀምራለን። ጥያቄ ያላችሁ ካላችሁ መጠየቅ ትችላላችሁ። ጨርሰናል። እሺ ጥያቄ ያለው ሰው is the role of adiposity in lipid metabolism? Yes, adipose tissue. Role of adipose tissue in lipid metabolism, it is storage only. This is for the white adipose tissue. For the brown one, it is thermogenesis. There is heat generation by the brown adipose tissue. White adipose tissue, it is for storage. We store lipid in the form of triacyl glycerol. እሺ ኔክስት ሌላ ጥያቄ ካላችሁ ሌላ ጥያቄ ያለው ካለ
አንድ ለደቂቃ ጠብቀን
እሺ
እስኪጽፉ ድረስ
እሺ What about carbohydrate metabolism, role of adipose tissue? Yes, in carbohydrate metabolism there is a role that is glycerol will be used for gluconeogenesis. Glycerol, as you know, it is one of the source in gluconeogenesis. It have a role in carbohydrate metabolism, especially during hypoglycemic condition. Could you make it clear the one about proenzyme? Proenzymes, they are secreted as inactive form. Inactive form means they are they are synthesized in the inactive form. They cannot catalyze the reaction. This is very important because number one, to avoid self-digestion, especially for protein enzymes, protein digestive enzymes. They can digest our own cell, like stomach. The stomach wall, it is again protein. There are different proteins. Those protein can be digested by those enzymes. ስለዚህ they become inactive because to avoid self digestion. Number two, to have a safe transport plus storage. Storage of those enzymes, it is occur as a result of making those enzymes inactive. እሺ it is very important. Activation of enzyme በሶስት አይነት መንገድ ሊካሄድ ይችላል። አንደኛው ክሊቬጅ ነው፣ ፔፕታይድ ክሊቬጅ። some parts of peptide will be cleaved. As a result, active site will be open, then catalyze the chemical reaction or bind substrates and catalyze substrate to product. ሁለተኛው mechanism, it is by the phosphorylation of enzyme. Phosphorylation of enzyme will activate the enzyme. The third mechanism is allosteric regulation ነው ወይም allosteric activation ነው። Regulators bind on allosteric sites and activate the enzyme. እነዚህ በሶስት መንገዶች ኢንዛይሙን አክቲቬት ማድረግ እንችላለን። ኢንአክቲቭ ፎርም ሆኖ ግን ለምንድነው ሴክሬትድ የሚሆኑት? አንደኛ ሰልፍ ዳይጀሽን እንዳይሆን እና ሁለተኛው ሴፍ የሆነ ትራንስፖርት እና ስቶሬጅ እንዲኖር ለማድረግ ሲባል ነው። እሺ ሌላ? እሺ አመሰግናለሁ። መልካም ውሎ ይሁንላችሁ። ቻው